In this presentation, I am going to describe about sex-linked inheritance. See, Thomas Hunt Morgan in 1910 described the sex-linked inheritance pattern in Drosophila melanogaster. Actually, he observed a Drosophila male fly which possessed white eye, and he used this fly in making crosses, and by observing the F1 and F2 generations results, he could deduce that the gene causing this white eye mutation is located on the X chromosome, and that was actually the finding of Morgan, that he was able to explain sex-linked inheritance pattern. So I'm going to discuss this aspect in much detail. But before I take up certain cases, certain classes, let us have some additional idea about this aspect. See, genes which are located on X chromosome, which do not have their homologous part on the Y chromosome, are referred as sex-linked genes or X-linked genes. And the inheritance pattern of these genes and the traits which they cause differ between the two sexes, between males and females. The inheritance pattern that differs, because when we observe the inheritance pattern of genes located on autosomes, then we find that whether reciprocal crosses are made, that does not affect the inheritance pattern. Means whether the gene is coming from the male individual or female individual, that makes no difference because they are situated on autosomes. But there will be difference if genes are located exclusively on the X chromosome or on the Y chromosome, and particularly those genes which are not homologous to the part of X and Y chromosome. And we can also observe the inheritance pattern of such genes in other organisms, including humans. And in our case, the color blindness and hemophilia are two well-known examples which follow the pattern of X-linked inheritance. Now, here is a particular class. In this class, you can see that how sex-linked genes get inherited. But simply I am showing the cross which has been made between Drosophila, two sexes of Drosophila flies, and the results which have been obtained in F1 and F2 generations. Its further analysis will be seen in further slides or next slides. See, a red-eyed fly, and that one is female, a red-eyed female fly, is crossed in this case with a white-eyed male fly. And in the next generation, all the flies, whether males or females, they possess a red eye. It means this red eye color is dominant over white eye. So it is just like the inheritance pattern we observe in cases of autosomes. Up to this level, you can see that red eye is dominant over white eye, and all individuals in F1, they show normal phenotype. But when these individuals are crossed with each other, then in F2 generation, the ratio of red and white-eyed flies, that definitely comes 3:1. But there is a peculiar, pattern, and that pattern we have to notice, that all females were actually red-eyed, or you will, if you perform this experiment, you will be getting all females having a red eye. So females will be red-eyed, and if you observe the male individual, you will find that 50% of the male will be red-eyed and 50% will be white-eyed. So exactly the same thing Morgan actually observed in his cross when he crossed white-eyed male with the red-eyed female. Then exactly the same inheritance pattern he observed, and he was really astonished to see that white-eyed flies were only male individual. So definitely he analyzed it to much more detail, and he was able to deduce that the gene causing this mutation, white eye mutation, is actually located on the X chromosome, and only in such situation this kind of inheritance pattern will be observed. Let us see how this happens. See, in this diagram, these two vertical lines which are equal in size show the X chromosome. Means a female fly will be XX type, a male fly will be XY type. So here two X chromosomes are shown, and in case of male, one X chromosome and other Y chromosome, which is quite short in size, that one is shown. So male is XY. If you see the locus where the allele deciding red eye or white eye are situated, then a female with red eye, that is indicated as W plus W plus. W plus allele is for normal eye color, that is red eye color. And this W that indicates allele for white eye color. So since it is a white-eyed male, the genotype of this male, means X chromosome, will have W allele on that locus, where in case of female it is W plus W plus. If you will cross these two flies, what will happen? This W plus containing X chromosome will combine with this W coming from the male, then it will be W plus W plus. So all females will have W plus and W genotype, and they will be red-eyed because W plus allele is dominant over the other one, recessive one, that is W. Whereas the male individual, will also be red-eyed because female will contribute its X chromosome with W plus, and that will combine with the Y chromosome, so male will also be W plus. But here the F1 female will be carrier. She will carry the normal allele as well. She will carry the mutant allele, but she will be of normal phenotype, so she is carrier. And when these F1 flies will be crossed with each other, then what situation will happen? What will occur in the F2 generation? You can see here. This W plus containing X chromosome will combine with W plus. So this female will be W plus W plus. Okay, so this will be red-eyed female. Now the other W containing allele that will combine with W plus. So this will be a carrier female but red-eyed female. So W plus W, heterozygous, red-eyed female, and to that I am referring as carrier. Then it is W plus containing X chromosome that combines with Y chromosome, so this will be a male individual and a red-eyed male. Then it is X chromosome with W allele that combines with Y chromosome, and this will be a white-eyed male. This is the reason that all males, if observed, if you consider only male individuals, then 50% of the male will be red-eyed and 50% of the male will be white-eyed, whereas all females will be red-eyed females. This was the result which Morgan actually observed, and exactly the same finding you will be observing if you will perform such experiments. And here in zoology department, Banaras Hindu University, we demonstrate such experiments to our BSc and MSc students, and exactly means the same result they get, and they actually analyze such results. So what kind of analysis could be done, that I am also showing in the next slide. See the F2 results that is shown here. as I told, suppose exactly the same experiment has been performed, and the red-eyed females, they are 510 in number. Red-eyed males, they come to 16 number. White-eyed males, they are obtained 230. Total becomes 1000. So 1000 flies were actually observed in F2 generation, and their number, that is shown here. So what you expect? You expect all females to be red-eyed. So 2 by 4 means half will be the number of females, and all will be actually red-eyed. So 2 by 4 you expect to be red-eyed, and that 2 females. Then 1/4 out of entire a number will be male individuals, and they will be red-eyed. So that number means that ratio you should get. White-eyed males 1/4 also. So out of 1000 you expect 500 to be red-eyed females, 250 to be red-eyed males, 250 to be white-eyed males. And if you get this expected number, observed number, you have already obtained, so you can apply chi-square test. I have talked chi-square test in so many presentations. That will be performed by using the formula summation observed minus expected, its whole square, divided by expected. So when you will apply that formula, you will get the chi-square value for these three different classes, and that value is shown here: 0.20, 0.40 and 1.60. These are the chi-square values for these three classes. Total comes 2.2. Since there are three classes, so degree of freedom will be 2, and table value at 2 degree of freedom at 5% level will be 5.991. So you have observed that your calculated value is quite less than the table value. So you accept the null hypothesis, and probability will be more than 5%. And you have actually decided that the null hypothesis is flies in F2 will be in the ratio of 2:1:1, where 2 will be the number of red-eyed females, 1 will be the number of red-eyed males, and 1 will be the number of white-eyed males. So in the present case, you actually accept that the F2 segregation pattern is in the same ratio as you wish, as you actually expect. Then one can also perform the reciprocal cross. In case of a reciprocal cross, you see, you have to take a white-eyed female, and that female will be crossed with a red-eyed male. Reciprocal cross means it is just an opposite situation. In the earlier case, you took a red-eyed female and that was crossed with white-eyed male. But here you will be crossing a white-eyed female with red-eyed male. So in the next generation, you will be getting red-eyed females and white-eyed males. So this means this kind of situation will be there. That is, in the F1 generation, in the next generation, all the females observed, they will have red eye color, and all males will have white eye color. And when these flies, F1 flies, will be crossed among each other, means with each other, then in the F2 generation you will find red-eyed females, white-eyed females, red-eyed males, and white-eyed males, and their ratio will be 1:1:1:1. That is, out of entire number of flies observed, almost 50% will be female, 50% will be male, and those 50% female will have half of them red-eyed, and for half will have white eye. Exactly the same situation will be there in case of male also. So this aspect you can see in the next presentation. Here in this diagram, see, there is a female. She will have 2X chromosome. A male will have 1X chromosome, other Y chromosome. So in case of a female, since this female is white eye, so the recessive allele will be present on both the X chromosomes. So WW alleles are shown. In case of male, Since this male is red-eyed, so W plus allele is shown because this one is a dominant allele, so it will be expressed here. Now, in the next generation, what will happen? This X chromosome with W containing allele will combine with W plus coming from the sperm. So the female will have W plus and W allele. So all females will be red-eyed because this W will also combine to this W plus. So females with the XX chromosome will be carrier. They will have one normal allele or the recessive allele. Whereas male, they will have W means mutant allele plus Y chromosome. So this W containing X chromosome combines with Y chromosome. So all males will be white-eyed. When you will cross them, then you can see that how there will be fusion between different kinds of gametes. This W plus containing X chromosome will be fertilized by a sperm with W containing. So it is W plus and W. This will be a female with red eye phenotype. Then another, you see this W containing X chromosome combines with W X chromosome of male and becomes WW. So this will be a white-eyed female. Now it is W plus that combines with Y chromosome and that will be a red-eyed male. Then it is W that combines with Y chromosome and forms a white-eyed male. So here you find a red-eyed female, then white-eyed female, then red-eyed male, and then white-eyed male. And the possibility of getting all these four different types will be completely equal. So they will be obtained in the ratio of 1:1:1:1. And if you have performed exactly the same experiment and suppose you observe the number of flies in F2 as the number is 205, means you performed this experiment in which you took white-eyed female, crossed it with the red-eyed male, and then in next generation you got all females red-eyed and all males as white-eye. Then those F1 flies were crossed with each other, and then in F2 you observed 205 flies which were red-eyed females, 190 as white-eyed females, 210 as red-eyed males, and 195 males which were white-eyed. Total, that is 800. So what you expect? You expect that is 1/4 will be female with red eye, 1/4 female white-eyed, 1/4 red-eyed males, 1/4 white-eyed males. So accordingly, you expect that 200 individuals will be for each class. So 200, 200, 200, 200, and their number will have to be exactly equal. Now you can here also perform this chi-square test to see whether significant difference exists between observation and expectation. So for this purpose, you will be applying chi-square test, and here the chi-square values which will be obtained for each class are given. Their total comes 1.25. Degree of freedom, since there are 4 different classes, so degree of freedom will be 3, and table value at 3 degree of freedom at 5% level will be 7.815. So here you find that your calculated value is very much less than the table value, so you accept the null hypothesis. Your probability is more than 5%, and you accept that the F2 ratio that follows 1:1:1:1 segregation pattern. So in case of sex-linked inheritance, particularly this experiment could be performed at a very large scale utilizing Drosophila as model organism, and there are a number of sex-linked genes. Their mutant types are there, and such types means any one of them could be used to perform such kind of experiment, and you would be getting exactly the similar pattern of similar inheritance pattern. The flies obtained in F1 and F2 could be analyzed, and the data could be explained exactly in the same manner.